Okay, here we are again, back again from lunch. So I hope you've had a good opportunity to use the facilities available to you on the platform and maybe got some lunch in and a cup of coffee as well, if you're lucky. Get us rolling this afternoon. We're going to be talking or hearing about molecular Lego, which I'm quite excited about. So with that, Daniel, over to you.
Cool. Thank you very much. So, hi everyone. I'm Dan. I'm a co-founder and CEO of MarraBio. We're a spin-out company based in Newcastle in the northeast of the UK. And yeah, I will be talking today about molecular Lego for cultivated meat and how to use that to engineer affordability at scale. So, if you're trying to grow cells outside of the body, you need to sort of trick them into thinking that they're in their natural environment. So, this involves giving them things like the right temperature, the right pH, some oxygen, and the right nutrients. But also, particularly in serum free media, it's important to make sure they get the right biological cues so that they grow and behave as you would expect. And the way people usually do this is to supply the media with a cocktail of usually recombinant bioactive proteins. And these are things like growth factors like FGF2 and TGF beta. But also for adherent cell culture, you also need something for the cells to stick to, which tends to be extracellular matrix proteins like vitronectin, laminin, fibronectin, etc. But the problem with these bioactive proteins is that they can be very expensive. They're often unstable with quite short half lives, and this makes them very difficult to use at scale. And this is particularly true in the case of cultivated meat. So the industry is obviously trying to develop products that can compete with traditionally farmed meat. But this is very challenging to do when you have a sort of cost of production that's over $20,000 per kilogram. And the majority of this cost comes from the media components, in particular the growth factors. And so, if we were able to find a way of reducing these costs, we could potentially get them down by about 99%. And this would address a really large and urgent problem in the industry and particularly accelerate the technologies towards the market. But for a problem of this sort of scale, it's not sufficient to just make the growth factors cheaper. We also have to look at whether we can innovate the way in which these bioactive cubes are supplied to the cells in order to actually reduce the amount of growth factors that are going into the process as well. So in that way, we really need to look at the bioprocessing of this and find new ways of doing things. So our approach to this is to use a protein that we've developed called cytocaf. And cytocaf is a bacterial protein. We express it recombinantly in E. coli, and it's quite a small protein. It's 15 kilodaltons. It's about 2 nanometers wide by 6 nanometers long. And what's really unusual about the cytocaf proteins is that they actually form into these very long, non-covalent, and highly flexible modular protein polymers. And these polymers themselves have a number of very useful but very unusual properties. Firstly, they're inherently biologically inert. So this means that mammalian cells don't really bind to them or stick to them or interact with them in any way. They're also, considering they're not chemically linked to each other, incredibly thermostable with melting temperatures above 80 degrees, which makes them very easy to work with as well as to ship and to store. And finally, we can produce them in very high yields from E. coli, meaning that we have the option to be able to produce these at very large scale and very low cost. And so the majority of what we are doing then is saying we've got a brilliant blank canvas scaffold for protein engineering efforts. It's structurally very simple, it's incredibly stable, and we can make a lot of it. So, can we use this scaffold to be able to create mimics of the bioactive proteins that are currently used in cell manufacturing? And the way this works, the core of the technology, is that we identify a factor that we'd like to try and mimic. In this case, this is a growth factor FGF2 that you can see. And you don't need the entire protein to be able to interface with the cells. In fact, you usually only need a small bioactive segment of this protein, a few residues as a peptide that can bind to a cell's receptor. So, what we do is we're able to identify these using literature searches as well as some other techniques. And we take this bioactive peptide motif and we genetically graft it inside the cytocaf gene sequence. And what this means is there's no need for chemical modification, and the bacteria will then just make the modified protein. And as you can see, the actual modified protein is pretty similar to the original. It's only a small modification, meaning that we've completely changed the bioactivity of this, but the other properties of the cytocaf remain the same. Its ability to form polymers, its ability to be very highly stable, are not much altered by the addition of this peptide. And of course, it still forms these very long polymers. So, at the end of this process, what we have are mimics of these bioactive proteins, but in this very sort of long, highly stable, animal free polymeric format. So, the Lego comes in because this is a non covalent modular polymer made out of little subunits. And what happens is, if you do actually heat these polymers above their melting temperature of 80 degrees, the polymers, they denature. And of course, because they're not chemically linked, they fall apart into the individual subunits. What's unusual about the cytocaf, though, is if you cool the solution down, these subunits actually begin to refold and self-assemble back into polymers again. And this gives us the opportunity to be able to mix different sets of subunits that have been functionalized in different ways, different building blocks, if you will. That, for example, one could mimic a growth factor, one could mimic an ECM protein. And we can use this technique to be able to combine these into a single multifunctional bioactive material. So how are we approaching reducing costs then? It's not just about making, a cheaper protein more scalably, although that is something we can do. But actually, what we want to do is utilize the unusual properties of this molecule to be able to attack the sort of costs involved in bioprocessing through many different angles at once. By having an ultra-stable protein, we can reduce wastage because we're moving away from very short half-life materials to things that can persist for a long time, in your cell culture flask or in your continuous process. We can reduce the number of inputs that go into the process. So rather than having this very complex cocktail made of lots of things, you can instead have a single material that supplies the bioactivities that your cells need. The cytocaf can attach to surfaces, meaning that it's not lost during every media exchange, every time you want to exchange for nutrients. And as I'll touch upon later, the high stability and the long length of these polymers actually permits them to be recycled and recovered from spent media as well and reused in another batch. Finally, this bioinertness also means that the cytocaf materials can have this quite unusual non-stick property. And in this case, we've been able to, as I'll show later, develop this to actually work as an anti-clumping reagent and sort of lower the aggregate size and increase cell yields from suspension culture. So I'm just going to take you through a few examples now of how we've been using the cytocaf materials in different ways. The first one is I'll show you about how we've been using it to make mimics of extracellular matrix and growth factor proteins. So we'll start with building a vitronectin mimic. So when cells bind to the extracellular matrix protein vitronectin, they do so using their integrin receptors. But even though there is a full vitronectin protein there, there's actually only about sort of 3 or 4 residues that these integrin receptors actually stick to. And this is the sort of core motif. So what we've done is we can transplant that from vitronectin into the cytocaf gene, express a modified protein, and now the integrin receptors on a cell can bind to that motif in exactly the same way, but in this time as part of a cytocaf scaffold rather than the vitronectin scaffold. So to test this out, we partnered with Dragon Bio, who are a manufacturer and seller of modified immortalized cell lines for cultivated meat. And we took their pig iPSCs and basically wanted to see whether they would grow on our vitronectin and laminin cytocaf mimics. And compared to recombinant vitronectin, we found that the iPSCs were able to attach and they were able to grow in a very similar manner to that they would on the actual sort of full-length original protein. So that's great because it shows that we can actually make mimics of these ECM proteins. Of course, these are stem cells, so we wanted to make sure that they retain the characteristics that you'd expect. And using immunofluorescence, we could see that they still retain their stemness markers. And also through embryo body formation assays, we could see that they retain their capacity to differentiate into the different germ layers too. So in this way, we've made just taking a small section of vitronectin and putting it into cytocaf, fully functional mimic of this particular ECM protein. But we wanted to go further than that as well and see whether we can do more complex bioactivities than just, attaching cells to a particular substrate. So we developed as a kind of proof of concept a mimetic of insulin, where it's a small peptide from insulin transplanted into the cytocaf backbone. And as you can see here, what we've been able to do is take some human MSCs and see them beginning to differentiate down an adiphogenic cell lineage, as you can see by the little patterns of fat droplets that are beginning to appear in the cells. And the crucial thing to remember here is, although the cytocaf proteins will work in solution as well, like traditional growth factors would, the key advantage here is by actually attaching them onto the surface and having the cells kind of come towards the growth factors and be in close proximity instead, so that they're not lost on the mediate changes. We then went further and did some work with the cell and gene therapy catapults. And our aim here was to look at growing iPSCs, and they can be grown in E8 media, which can and when scale up can be very expensive. And our idea was, can we introduce a cytocaf, a TGF beta cytocaf mimic that's able to take one of those growth factors floating in the media and immobilize it on a surface and still get the same results in a simpler media form? And the sort of short answer is yes, we were able to do this. We used our vitronectin cytocaf to anchor the cells and then grew them in either E8 media or E6 media with the TGF beta cytocaf also on the surface. And what you can see is we retained the pluripotency markers expected with the cells, and we were basically able to remove this from the media and attach it to the surface instead. So to follow up on this, I think it will be quite exciting to see, can we then cause a shift from more expensive, more complex medias to using simpler medias with instead a very ultra stable kind of immobilized substrate providing those bioactivities instead. And just as a demonstration then, it's not so relevant to cultivated meat, but to show some work that we did a few years ago with the mosaic polymers, we were able to create a mimic of osteopontin and BMP2, put them together into the same multifunctional material, attach this to a surface and grow again human MSCs, and use this to trigger the differentiation of the MSCs down an osteogenic lineage. And you can see the patches of mineralization there and also changes in gene expression over a culture time of about 2 to 3 weeks. So in this way, again, we didn't need to add any growth factors into the media. We could rely solely on what we'd immobilized on the surface in the cytocaf backbone. So moving on from them to suspension cultures, again, trying to utilize some of the unusual properties of this protein, we wanted to see whether this sort of bioinert non-stick nature could actually be useful as a sort of a physical way of stopping aggregates from clumping together too much. So to do this, we again partnered with Dragon Bio and they used their pig iPSCs for this. This time they suspension adapted the cells, and you can see when growing in aggregate and suspension, they still again retain their pluripotency markers. And so what we wanted to see was, if we add the unmodified cytocaf in this case into the media, can it act as a sort of very benign physical barrier to stop these things from actually sticking together and clumping so much? And as a result, can we get more cells? And what you can see is that in the presence of the cytocaf, we were able to get more smaller aggregates. And in the end, that resolved into having about a twofold increase in the number of viable cells from the culture volume. So in this way, we can use the cytocaf not just as a mimic of things, but as a reagent that can work to help sort of declump and actually increase cell yields from suspension culture as well. And we're now looking into whether this can also work with microcarrier-based formats too. So finally, I think one of the other ways in which we can reduce the amount of these reagents being utilized in bioprocesses is by actually reusing and sort of recycling some of these materials. Now, the cytocaf, as I've mentioned, it forms very long polymers and it's incredibly highly stable. So it really is sort of almost perfectly designed by nature to be able to be recovered from spent media and reused. And so we wanted to see if we could generate a proof of concept of this. And to do this, we took some of our fibronectin cytocaf, we took some media and spiked the cytocaf in. We sort of left it overnight and then put it through a high molecular weight cutoff filter. The idea being that any of the small components of media, any waste things, any nutrients, anything secreted by the cells would pass through the filter, whereas the cytocaf, being very, very large, would be retained. And actually, by doing this, we were able to recover almost all of the protein that we'd originally put in, which was exciting. And from the spectra, it didn't really look, the stuff we'd recovered didn't seem to be much different than the stuff we put in. But then the key question was, could we then use that sort of recovered and reused material to actually grow as a batch of cells? And so again, as proof of concept, we looked to human MSCs. We took our sort of fresh fibronectin cytocaf and coated a surface with it, and we're able to see the MSCs growing, attaching and growing onto that substrate. And then we compared this with the material that we'd recovered from the media, and actually we saw that they performed almost equally, with the cells looking the same and with similar numbers of cells on both. And so with this, it's a first step, we believe, towards being able to show that we can actually have a reusable material that can either be used for multiple batch processes or potentially in continuous processes too. So just to sort of round off then, hopefully what I've shown you is we've got this quite unusual but quite sort of intriguing material, which we can produce an awful lot of, and we can also use it by using protein engineering techniques to develop mimics of the bioactive proteins routinely used in the manufacture of cells for cultivated meat. But the key difference is being that this is a very stable protein, a very stable backbone, so you don't lose so much of the material over time. You can use this Lego-like function of heating and cooling to be able to build more complex materials through a very simple process. You can stick it to surfaces or use it in suspension, but if it's stuck on surfaces, then at least you don't lose the protein every time you do a media change. And if it is in suspension, it looks like you can also recycle the protein and reuse it in a second process. And finally, by utilizing the sort of non-stick nature of a protein, we can actually develop it as a anti-clumping reagent as well for cells in suspension in order to boost the yields of those cells. So we're currently interested in looking for beta testers, particularly for the anti-clumping reagents and some of the growth factors. And if you're interested, please feel free to get in touch. The application note with Dragon Bio, you can find the link to there through LinkedIn, and also there's a QR code for our contact info there. So thank you very much for listening. I hope that was interesting, and I'd be happy to take any questions.
Great stuff. Thank you. I mean, fascinating, really. I was watching the bit about, sort of melting them down and then coming back together, and it put me in mind of that thing with the sea sponge in the blender where it sort of makes itself again. It's remarkable what some of these things can do. It plays with you. But anyway, to questions, straight into a question from the audience, if I may. How straightforward is it to recycle cytocaf? And is this process scalable?
I think the answer to that is both. I mean, obviously, the data we have is just with a small scale lab pilot at the moment, but the way we did it was using a vibro system from Sani Membranes. It's essentially the technique is tangential flow filtration. So you've got this big membrane. It's got quite a high, I think, molecular weight cutoff, and you basically flow the solution on there. It shakes, anything small goes through, anything big gets kind of put back into your original container, with the volume reducing over time, allowing you to concentrate it. So it was very easy to do because you just took the media and just put it into this machine and waited for it to come out. I'd hesitate to say it's automated, but you don't have to sit there with it while it does this. And of course, the key about scalability, they've built this machine to be scalable. So we've got just a small benchtop one, but they go up way to, I don't know, liters. These things are the size of rooms. So I think provide with a bit more development and sufficient interest, then I believe it could be quite a useful thing to have. And particularly thinking about perhaps some of the things I've seen with continuous processes where you've got kind of media constantly flowing. I don't think it would be too difficult to then just add in this extra machine where the waste media is flowing into that and it's automatically concentrating and recovering this material. So early days, but I think the answer is yes.
Nice. Okay. Picking up on this theme of the sort of modularity, the Lego approach, if you will, um, how does that enable kind of the customization for different cell types? And like, what's your process for designing a new module to go
Mhm.
with that?
Yeah, so at the moment, it primarily, if someone was to come to us and say, "Can you make X, Y, or Z?" We usually first conduct a literature search because people have had a look at, trying to reduce these growth factors or ECM proteins to peptides, or identifying the bit that receptors bind to over many years. So there's quite a lot of information out there already. We have several different positions which we know in the cytocaf are really tolerant of these kind of insertions. So we tend to put it in one or multiple of those and then test the expression. When we are able to produce that, that means we've got a new module. The whole process of that takes maybe sort of about 2 to 3 months, depending on kind of whether anything goes wrong and whether how quickly the various reagents arrive. But once we've got that, it's into biological testing. I think in terms of customization for cell type, the thing that we're really interested in at the moment and kind of following up on is taking, we've got I wouldn't say it's extensive, but we've got a fairly large catalog of some of these things we've already built, and it's can we then use this Lego-like approach to combine these things to make bespoke combinations of these modules that are geared towards different cell types. So, for example, we've got a project looking at growing these for endothelial cells with the sort of VEGF growth factor. You saw the work with the catapult there, where we were trying to create a sort of vitronectin FGF2 TGF beta surface and things like this. So that's kind of, I think, where the next step is. We've got a lot of the modules already, and now we're at the stage where we're trying to combine these and see, how well they support the desired biology of the different cell types.
Right, thank you. Question here from in the audience. Thank you for your eye-widening presentation, which I'd agree with actually. Um, he wants to know if he's got this right. The presence of varying peptide inserts in the cytocaf monomers does not affect the temperature at which the polymer breaks up. Is that correct?
Not significantly. It affects it by sometimes like 1 or 2 degrees when we've measured it before, but the wild type material has a melting temperature of 86. So I don't think we haven't seen any that are really sort of below 80-ish really. So there is a small effect on the stability, but not any that would significantly alter the utility of the protein.
All right, fair enough. Excellent. Um, I mean, as I have to ask this, as much as it sounds, amazing in everything it does, what are the limitations of the system, particularly when it comes to sort of mimicking complex growth factors?
Yeah, good question. I mean, it's the main limitation we have at the moment is we are quite dependent on scanning the literature for the sequences that we want to plug in there. And so it's all well and good if someone's done the work, but if they haven't, then that's a bit of a challenge. The way we're trying to overcome that challenge is twofold. Firstly, we're beginning to explore some of these new AI tools, particularly centered around kind of protein design and structure, to see how we might design these new things in silico and test them ourselves. But the second is we've also got a more extensively changed cytocaf module, which basically contains binding sites for things like FC tags, biotin tags, and other type of tags, which means that you can plug and play third-party growth factors. So if it's one we can't make a mimic of, you can still actually immobilize this within the CAF1 structure. And although it's the data we have on that is still fairly early, the preliminary indications are that still helps reduce the amount of growth factor you need by sort of sticking it into a scaffold, immobilizing it on a surface, and again, not losing it or adding it as frequently as you might do otherwise.
Okay, interesting. And I guess one last question, if I may. We're all very conscious of this. I've, mentioned it a few times over the course of the day with various speakers. Are there kind of regulatory or legislative hurdles that you see here with this, in deploying this in the sort of commercial systems, and what are those hurdles?
Yeah, it's something I think we always have to ask ourselves, and obviously making using engineering biology techniques, you're always making kind of new things, and it might always be it's whether the regulations are kind of a bit behind the curve on where some of the technology is. So I don't have a definitive answer. It's still a little unclear to me at the moment. But what we've tried to do is approach the problem head on, particularly by partnering with CPI and a company called Alias Biotech. Both those organizations are here on the conference today. So if you want to ask them more about what they're doing, you can. But what we did, we worked. You might have seen Jonathan Rand's talk this morning. We worked with his team to do HACCP analysis and have a look at the basics of how does our production process match up with what you need to get to a food grade standard. So that was looking at the quality of the production and how you make something that could meet those requirements. But I think beyond that, looking at the FDA approvals of the first cultivated meat products in the US, we wanted to see what happens if you were to ingest and digest this protein, because obviously if it's something a bit different, it's come from a bacteria, people don't tend to eat it. So it's what does that mean? And so to do this, we worked with Alias, who have a model of the human gut system, and basically simulated the digestion of this to various different levels and different degrees. And we're basically able to see that it gets just digested like any normal protein would. So we didn't see any effect, for example, on the gut cells that they had in their model, which was really good because that would imply that if you ate it, you just digest it like anything else you might encounter. So we've got our next step is then to talk to regulators and the FSA and things to see what they make of this data. But that's how we've tried to kind of get in front of some of the issues we think we might get asked.
Yeah, I mean, it's fair enough. You've just got to do what you can, but that's interesting to know that you are already looking to that. I think that's very wise. Um, we're going to wrap it up there, but Daniel, thank you so much for joining us today and for that eye-widening presentation. I like that. I'm adopting that as an official phrase. Um, yes, thank you for joining us. If you have further questions for Daniel, do reach out to him via the platform or elsewhere. And yeah, we will be back with our next speaker very shortly.
Thank you.
Nice.